Now I'm going to show you several different manipulatives that are going to show you just how versatile the cell membrane is. The first structure that I started to build here is the outer membrane of a typical eukaryote cell and just the outer phospholipid membrane. We're going to add some proteins to it in a minute. So I'm just going to finish that quickly by putting what we call one of the curved pieces up to give it that cool look, and then place the membranes on the top. Now your students at this point are going to be drawing the cell membrane. Now the instructions that you need to give them at this point, what I do, what some of my colleagues do, is we don't expect them to draw every phospholipid, right? So they would draw maybe 8 or 9 phospholipids, and then after that, for a rough note, they can just go back to the old school align, and we can give them some instructions later on if they're going to do an assignment. But they certainly don't have to draw all the phospholipids. So here we have an outer membrane. Using your 8 other curves, I'm going to draw, and then I was going to say sketch. I'm going to build 2 structures. The second one that I built here that's sealed, I'll put the enzyme in it. So this could be a lysosome. This kind of structure here can represent like nuclear pores, and it's a simple diagram of a nucleus because it doesn't have the second membrane, but that's okay. So we can have a nucleus here and a lysosome, and now I'm going to add some proteins to the membrane. So you can tell from your legend that's a receptor, and here's a glucose or protein channel, right? That could be either or. It's a channel nonetheless. Now, you have other proteins in your kit that I call a generic protein, and we're going to use those for different proteins, which I'm going to start to build right now. One is, we're going to add one more surface protein. So here's 3 other proteins that are in your kit. I'm going to take the 2 generic proteins, and I'm introducing even my grade 9s to an identity. I call this an identity marker, and that kind of structure is fine. So really, I'm leading my grade 9s and grade 11s into MHCs by the time they get into grade 12. So we've added receptors, channels, and identity marker onto the surface of the membranes. Now, the other thing you can do with the proteins is you can then take your proteins and take this type of structure and assemble them in some creative fashion and build what we would call one section of cytoskeleton. And you could have the students sketch the cytoskeleton in whatever position you like. The other very interesting thing that created with the proteins is you can continue this theme of a cytoskeleton, all right, with these guys, right? You can then take this structure like this and like this, and grab our enzyme now. And this is our classic structure here that we like to call, so see the little man here? 2 legs, here's his body, and that's kind of his head that's carrying a vesicle, and let's put something in the vesicle. Now, that worker protein here is going to travel, right, along the cytoskeleton and move the vesicle as travels on the cytoskeleton. So you can play with that and create a very cool image of a worker protein. The next thing I'd like to build for you is the mitochondria. Now, to build the mitochondria, we now remove ourselves from the cell membrane and I'm just going to use the pieces to build the inner and the outer membrane. One thing I'd like to give you from a directional perspective, obviously you're building different pieces now of the cell. Maybe what I just showed you is one lesson. The next day you come in, you build the mitochondria. Like what my students would do is take smaller pictures of those structures, and then if you can remember seeing, excuse me, the grade 11 image of the final assignment of a cell, they would collate all those images into one cell. So they would now take a diagram of the mitochondria. So to build the mitochondria, we're just going to, we have an outer membrane here now. So we just are going to build an inner membrane. And here's your cristae. And I need one more piece from here, right, like so. And I start to cheat a little bit here, obviously, but hey, that's okay. So now we have an outer membrane and an inner membrane for the mitochondria. And again, you can put some proteins in. Certainly if I was teaching grade 11s, I would put some proteins in here. If I was teaching grade 9s, I'm introducing to them that there's two membranes here. Now for a chloroplast, as you know, there's three membranes to a chloroplast. So I'm not going to build the outer membrane. What we're now looking at is the inner membrane, and I'm going to build two grana, like so. So there's one, and there's two. So again, depending what level you're at, right, we now have the inner membrane, and here's our thylakoid membranes represented by two grana. So that's a very good representation of a chloroplast, and you can begin to understand the power of the membrane showing all these structures have the same phospholipid bilayer. You do something very just simple like this. I fuse these together, right? I remove these two pieces, and now we got a Golgi. So there's a picture of a Golgi. We move from that to the ER. Now the ER is a little bit different. So I will take these pieces here and go like so. Here and here. So what I'm building here now, when we build our ER, you know that the outer membrane, this is actually the outer membrane of the nucleus, actually starts to fold in, and that outer membrane actually starts to become the membrane of the ER. So this is now the ER part, and then we take a curve, go up like this, and over to here, and we go like this. And this gives us a picture of our ER. So that's a nice picture of the ER, and what can you now build on top of that to finish the lesson? Well, you can start taking these proteins and build a ribosome, because that's where ribosomes hang out. And whatever kind of creative mood you're in, there's a ribosome hanging out on top of the membrane of the ER. And those are all the structures that was my idea to show you in this particular lesson of what we call basic cell structures. So I'm sure you can create some more, and you can have a lot of fun with it, enjoy it, and let me know what new ones you've created.